This is the outline for the presentation. I tried to cover the highest yield principles of these 12 lectures. A total of 21 questions. We're going to discuss them, the principles they include. Okay, starting with the first question. If a sample of a DNA contains 35% cytosine, what is the percentage of the DNA base pairs are thymine? A, 15%, B, 20%, C, 30%, and D, 35%. The answer is A, correct. So this is very easy question according to the Chargaff's rule, where the indicates the percentage of adenine is equal to the percentage of thymine, and the percentage of cytosine in the DNA is equal to the percentage of guanine. So basically by duplicating 35 plus 35, and then subtracting it, dividing it by 2, you'll get the percentage of thymine and adenine. So thymine and adenine would be 15% each. Okay. So that was a straightforward question. Moving on to the next question. An 8-month-old child is brought to the pediatrician office due to excessive sensitivity to the sun. Skin areas exposed to the sun for only a brief period of time were reddened with scaling. Oh, sorry. Irregular dark spots have also appeared. The pediatrician suspects a genetic disorder in which of the following processes? D. Excellent. Xeroderma pigmentosum is the diagnosis. So yes, it is the nucleotide excision repair. So this is a very high yield information. The xeroderma pigmentosum is a very important disease to remember. So normally, under normal condition, UV light induces the formation of thymidine dimers. Also, we call them pyrimidine dimers, which are normally excised by the nucleotide excision repair mechanism. In xeroderma pigmentosum, it is an autosomal recessive disorder, which is caused by defective in the nucleotide excision repair. This will lead to the accumulation of these pyrimidine dimers, which causes the skin freckling, reddening, and ulceration, and ultimately can lead to skin cancer. So knowing the mechanism of the nucleotide excision repair is important, is that we have an endonuclease that removes the damaged segment, and then it is replaced by DNA, polymerase, and ligase. So reviewing the two of the most important DNA repair mechanism, the first one is as we mentioned, the nucleotide excision repair. We're an endonuclease. So this is really important, a distinguishing point to remember. When we're talking about the nucleotide excision repair, it's the endonuclease that remove the damaged bases, which are the pyrimidine or thymidine dimers. And then the DNA polymerase fills the gap, and DNA ligase reseals the gap. When you have a question of defective nucleotide excision repair, it is defective in xeroderma pigmentosum, as we discussed in the previous scenario. Mismatch repair, it's another DNA repair mechanism where we repair the base mismatches, like if adenine is basing with guanine instead of thymine or cytosine, and vice versa. When the strand containing the mismatch is identified, the mismatched nucleotide is removed by an exonuclease. So again, this commonly tested that in the nucleotide excision repair is removed by an endonuclease, whereas the mismatch repair is removed by an exonuclease. And the mismatch repair is defective in Lynch syndrome, also known as hereditary non-polyposis colorectal cancer. The genes of the mismatch repair often start with MSH, MLH, and MSI genes that cause the Lynch syndrome, which colorectal cancer that are not associated with the colon polyps. Any questions for this? Okay, moving on to the next question. Okay. So, third question. 4 samples of DNA were analyzed for their melting temperature. Which sample is most likely to have the highest melting temperature? Okay, so you're already answering B. Excellent. Yes, so it is B. Again, this is a very straightforward question, the DNA melting temperature. So the higher the GC content, the guanine and the cytosine content, it leads to more hydrogen bonds between the bases, making the DNA double helix more stable, requiring the higher temperature to melt. So as you can see, cytosine and guanine, they have 3 hydrogen instead of the 2 hydrogen when compared to thymine and adenine. So thus the 60% makes it the highest percentage of stable bond, and thus the highest melting temperature. Okay. A 65-year-old woman. Exactly. Okay, moving on to the question 4. A 65-year-old woman came to the emergency department due to profound fevers and malaise. After the initial evaluation, she is hospitalized for septicemia, which is the infection of the blood, bacteremia of the blood. Blood cultures plated on lactose-containing media grow rapidly dividing gram-negative bacteria. Replication of these microbial cells require the synthesis of two daughter strands of DNA using the parent strand as a template. Which of the following processes will differ the most between the 2 daughter strands formed at each replication fork? So this is a long one. Let's divide it. Let's highlight the most important parts. So as we mentioned that here we're looking for the bacteria that the replication requires the synthesis of 2 daughter strands of DNA using the parent strand as a template, which is the same as the human. Yes, true. The second part, which of the following process will differ between the 2 daughter strands? So as a hint, you already answered that the answer is C, true. What we're referring to the 2 daughter strands here is the leading and the lagging strand. So at the this is the origin of replication, and we have the 2 daughter strands that use the parent strand as a template are the lagging and the leading strand. The difference between the leading strand and the lagging strand, as you can see, is the leading strand is the going to away from the origin of replication, and it is in a continuous manner. Whereas the lagging strand is the synthesized in Okazaki fragments that need to be joined by a DNA ligase. So the other things that DNA helicase, which is required to break down the replication fork, the same for both. Interaction with a single-stranded DNA binding proteins, which prevents the DNA from re-annealing, is the same for both. Proofreading is the same, and relief of supercoils by topoisomerase is also the same. So the only difference is that leading strand does not require joining of the fragments by a ligase. Excellent, yes, sir. Okay, so we already explained this. Moving on to the 5th question. A patient suffering from chills, vomiting, and cramping was rushed to the emergency department. He had eaten wild mushrooms for dinner that he had picked earlier in the day. His symptoms are due to the inhibition of which of the following enzymes? B. Excellent, everyone. Alpha-amantine. Yes, excellent. It is the answer is B, RNA polymerase 2. So alpha-amantine is the patient most likely has ingested alpha-amantine, which is a toxin that at a very low concentration inhibits the RNA polymerase 2, blocking the transcription of the single copy genes. RNA polymerase 2, as you know, it makes the messenger RNA through the process of transcription of the DNA. And RNA polymerase 1 and 3 are more resistant to the effect of the amantine, and the toxin has no effect on telomerase or any type of the DNA polymerase. Okay, moving on to the 6th question. A researcher has isolated a complementary DNA for gene Y He performs the following Northern blot experiment using samples prepared from different tissues. A potential explanation for this finding is which of the following? So A, alternative splicing, B, DNA mutation, C, enhancer effect, D, DNA rearrangement, and E, transcription factor effect. Correct. The answer is A. So as you can see in this, that we have the gene Y, which is the complementary DNA, and from different tissues, we have different protein kilobases, or they have different sizes. So what accounts that we have the same gene, but the different protein products is through alternative splicing. As you can see that here, the alternative splicing is the process where the exons of the premature mRNA is produced by the transcription, and then they're combined in different ways in order to produce different proteins. So it's clear in this picture that we produce the different protein isoforms. And this accounts for the the variety of troponins, which is found in the muscle and in the heart, the tropomyosin variations as well, and the difference of secreted versus the membrane-bound immunoglobulins. Now, splicing is defective in beta thalassemia and also in some types of cancer. So this is important to note that defective alternative splicing is seen in beta thalassemia. Any question? Okay, moving on to the 7th question. Okay, that's explaining why not the other. So DNA mutation would not do this because it would change the whole sequence of the gene. DNA rearrangement, it is the process that takes place mainly in immune cells, not in all tissues. And it results in the affinity of the immune cells. And the transcription factors, they influence the RNA polymerase affinity for specific genes by binding to the promoter or the enhancer region, which either stimulate or inhibit the gene transcription, but they're not going to affect the post-translational processing of a protein. Okay. So a 22-year-old woman sees her physician for a variety of complaints over the past year. These include fevers that come and go, fatigue, joint pain, and stiffness, and a butterfly rash on the face, which is seen in this picture. Sores in her mouth, easy bruising, and increased fatigue, feelings of anxiety and depression. A diagnostic blood test would show autoimmune antibodies directed against which class of molecules? A, perfect, acetylation. Yeah. Ribonuclear protein complexes is the answer. So ribonuclear protein complexes, these are the involved in mRNA splicing, where we know the process of the mRNA splicing is the removal of the introns, which are the non-coding sequences. They often start with GU and ends with AG, in order to form a mature mRNA. The splicing is performed by the spliceosome, which remove the introns containing a GU at the 5' end. So we start with removing the GU at the 5' end. As you can see here, first the small nuclear ribonuclear protein, they bind to the adenine at the branch point. This is going to attack the GU at the 5' end, and then forming this structure with a spliceosome. And this shape of the intron, we call it a layetate. Okay, so the layetate-shaped loop is the intermediate generated. And then the hydroxyl group that was removed here is going to attack the AG point, and then to remove all of the introns, the intervening introns. So the cleavage at the 3' side, then the layetate is going to be released, removing the introns and joining of the 2 axons. And antibodies against these small nuclear ribonuclear proteins, or SNRPs, are associated with SLE, leading to inability to form the spliceosome and cleavage of the pre-mRNA. Okay. A 2-year-old boy is being evaluated for failure to thrive and developmental delay. His past medical history is significant for recurrent ear infections since the age of 6 months. Physical examination shows coarse facial features, corneal clouding, fat splenomegaly, and restricted joint mobility. Mass spectrometry analysis performed on the cultured fibroblasts and reveals deficient phosphorylation of the mannose residues on certain glycoproteins in the Golgi apparatus. Normally, these proteins are most likely to be transported to which of the following cellular locations? Okay, so you answered B. That's correct. Lysosomes. Do you know the name of the disease? No, it's not Hurler. So the main problem here is the phosphorylation of mannose residues. Exactly, eye cell disease. So there is a deficient, it may mimic Hurler because of the corneal clouding and the restricted joint mobility, the skeletal abnormalities. But they mentioned that the problems are deficient with phosphorylation of mannose residues, which is seen with the eye cell disease. So eye cell disease, it is a lysosomal storage disorder. It is autosomal recessive. The enzyme which is defective is the glucosamine 1-phosphotransferase. This will cause the failure of Golgi to phosphorylate the mannose residues. So decrease in mannose 6-phosphate phosphorylation, what's going to happen is that these enzymes that should go into the lysosomes will end up to be secreted extracellularly rather than be delivering to the lysosomes. So the lysosomes deficient in digestive enzymes will cause the buildup of cellular debris, which can appear as the inclusion bodies. The clinical features are very similar to that of Hurler, especially for the coarse facial features and the corneal clouding and restricted joint movements, but they also have gingival hyperplasia, slow hand deformities, kyphoscoliosis, and labs which show an increased plasma levels of lysosomal enzymes. Why? Because that instead of being secreted, instead of being delivered into the lysosomes, so mannose 6-phosphate is acting like the signal that makes them go into the lysosome. If it is absent, they cannot go into the lysosome and they're going to go into the blood, which can be detected in the blood test as high levels of lysosomal enzyme. Okay. Moving on to the 9th question. Theoretically, a disease could result from an increased expression of a particular gene. This can occur in eukaryotes through a single nucleotide mutation in a promoter proximal element. This best explained by which one of the following? So the question is asking that if we have an increased expression of a gene occurring in the mutation near a promoter proximal element due to what? A, more efficient splice site recognition. B, increased opportunity for hydrogen bonding to transcription factor mainly. Beneficial amino acid replacement derived from the missense mutation. Increased amount of sigma factor binding or reduced energy needed to melt the DNA helix at this position. B. That's correct, it's B. Increase opportunity for hydrogen bonding to a transcription factor or a transacting factor. So why is that? Because going to the regulation of the gene expression, when we have a single nucleotide substitution in a promoter region, that causes an increase in the gene expression, it is basically because of increased binding of an activator or a transcription factor to an enhancer or a promoter region. This will lead to an increase in the gene expression. The sigma factor, which is mentioned, it is only in prokaryotes, and it does not code for an amino acid since the exons are not in the promoter region. And the DNA helix does not have to be significantly melted for this area to allow for the transcription factor binding. So it's basically a mutation in a promoter or an enhancer region that caused a transcription factor, which is demonstrated here as an activator to bind more avidly, and this thus increasing the transcription. Okay, any questions? Okay, now moving on to the 10th question. The acetylation of histones has which of the following effects? A, uncoiling of histone structure, preventing it from being accessed by transcription. B, uncoiling of histone structure, allowing it to be accessed by transcriptional machinery. C, coiling of histone, allowing it to be accessed by the transcription. D, coiling of histone, preventing it from being accessed by the transcription machinery. D. That's correct. The answer is D. Coiling of the histone structure, preventing it from being accessed by the transcription machinery. So this is really important to know that histone acetylation loosens the chromatin structure. So as you can see, the acetylated histones are, they lose the chromatin structure. So this is going to cause an activated transcription. So remembering that acetylation activates transcription, whereas deacetylation of histones make a tight chromatin structure, and this is going to decrease the transcription. So deacetylated histones, they have decreased the transcription. Yeah, exactly. It forms a heterochromatin. Okay. A 42-year-old woman, she comes to the neurologist for the enrollment in a research study. She has a 15-year history of resting tremor, bradykinesia, and cogwheel rigidity, which are consistent with the Parkinson's disease. One of her siblings recently started having the similar symptoms. Genetic analysis is performed on the patient and her affected sibling, and the result shows a loss of function of a mutation that leads to the accumulation of misfolded proteins. Which of the following biochemical processes is most likely to be defective in this patient? A. Yes, perfect. The process of ubiquitination. Yes. So acetylation, gamma carboxylation, all of these processes, they have nothing to do with the getting rid of misfolded proteins. But the process of ubiquitination, where it is involving the ubiquitin that acts like a marker of the misfolded or damaged protein, it attaches to them and then labeling them for degradation by proteasome. The proteins, then they enter the proteasome and they're degraded into the smaller peptides. Now, impairment of ubiquitin proteasome system, it contributes to the development of neurodegenerative disorders like Parkinson and Alzheimer's disease. Do you know what is the specific? Yes, exactly. Do you know what is the specific misfolded protein in Parkinson?
Alpha-synuclein?
Yes, exactly. It is the alpha-synuclein. So, and in Alzheimer's disease?
Beta amyloid.
Yes, beta-plated amyloid sheets. Excellent. Okay. Question 12. A 3-day-old boy is brought to the office due to poor feeding, vomiting, and progressive lethargy. The parents also report that his diapers have a burnt sugar smell, differing from the previous children's diaper. The mother is exclusively breastfeeding. Her other 2 children are healthy and have no medical problems. Physical examination shows a lethargic infant. dry mucous membrane, and generalized hypertonia. Which of the following should be restricted from the infant's diet? Phenylalanine, no.
Leucine.
Yes, perfect, leucine. Branched chain amino acid, yes. So the key here is the burnt sugar smell of the diapers. The choice D is tricky because you might think that it is phenylketonuria, but phenylketonuria, they have a musty odor rather than a burnt sugar smell. So it is the maple syrup urine disease, which is due to the defective of a branched chain alpha keto acid dehydrogenase complex. So in maple syrup urine disease, we have deficiency of BCKD, which normally causes the breakdown of the branched chain amino acids, which are 3: leucine, isoleucine, and valine. The deficiency is usually neurotoxic, causing hypertonia, poor feeding, lethargy, and irritability. And they have the characteristic of a sweet odor of the urine. This is different from the musty odor in phenylketonuria. And usually phenylketonuria would also have like skin eczema and hypotonia rather than hypertonia. Diagnosis confirmed by the presence of of the branched chain amino acids in the levels in the urine, or by analysis of the enzyme itself, absence of the enzyme. The therapy consists of the dietary restriction of the branched chain amino acid, but the patient remain at lifelong risk of neurotoxicity in the setting of intercurrent illnesses and fasting. Okay, question 13. A 2-year-old boy is evaluated for easy bruising. The skin is difficult to suture due to its extreme fragility. On physical examination, there is a hyperextensible skin, multiple ecchymosis over the forearms and pretibial region, and an umbilical hernia. A skin biopsy performed, and the histochemical evaluation reveals a defect in the extracellular processing of collagen. Which of the following steps of collagen is most likely to be impaired? This is a tricky question. So the main thing that the question is asking here is which step of collagen does occur out of the cell? So all of these are the are both translational modification of collagen, but the defect is in the process that occurs in extracellular processing, so not inside the cell. So glycosylation occurs in the cell. Interchange C-terminal disulfide bond formation within the cells. N-terminal propeptide removal occurs in the outside of the cell. So it is C is the correct answer. So looking into the diagram here, you can see clearly the process that occurs inside the cell and the process that occurs outside of the cell. Inside the cell membrane, we have here first in the rough endoplasmic reticulum, the first three steps, which is the pro-alpha chain backbone synthesis, hydroxylation of proline and lysine, and the glycosylation. Then we have triple helix formation. All of these three steps occurs within the cell. After exocytosis, then we have the cleavage of the procollagen C and the N-terminus, and that was the answer. Is that clear? So going back into the question, as you can see that the skin biopsy revealed a defect in an extracellular processing of collagen. So this is a rare form of Ehlers-Danlos syndrome. As you can see, as you may know that Ehlers-Danlos syndrome is due to the collagen deficiency or defect, but it may also be due to a procollagen peptidase deficiency, which is causing an impaired cleavage of the terminal propeptides in the extracellular space, as seen here by step 5. So the main question was asking you about the compartments where the processes happen. And it would cause the same presentation as Ehlers-Danlos syndrome. Is that clear? Okay. Question 14. Post-translational glycosylation in the endoplasmic reticulum is which of the following? O-linked glycosylation in asparagine residues, O-linked glycosylation on serine or in threonine, N-linked glycosylation in asparagine, N-linked glycosylation on serine and threonine, O-linked on glutamine or N-linked on glutamine. So we have two forms of the glycosylation. We have the O-linked and the N-linked glycosylation. Yes, exactly. B would be on the Golgi. So the answer is the N-linked glycosylation in asparagine. As we mentioned that we have the glycosylation are forms of post-translational modifications of protein. So we have the N-linked glycosylation. It occurs on the asparagine in the rough endoplasmic reticulum. And the O-linked glycosylation occurs on serine and threonine in the Golgi apparatus. Okay, so this is an important, straightforward question and commonly asked about the N-linked, the difference between N-linked and O-linked glycosylation. Okay, whether it is N and O-linked, where does it occur in the rough endoplasmic versus Golgi, and on what residues, on what amino acid, asparagine or serine and threonine. Okay. Okay, moving on to question 15. Tyrosine is not involved in the process of the glycosylation of the N-linked glycosylation. It's either on asparagine occurring on the N-linked glycosylation or O-linked on threonine, as you can see here, or serine residues. But tyrosine is not involved in the process of N and O-linked glycosylation. O-linked? I did not get the question. So the O-linked glycosylation occurs mainly on serine and threonine, and it is in the Golgi, serine and threonine. Either serine, threonine is seen in the picture, it can also occur on the serine. But the N-linked glycosylation occurs on asparagine and the rough endoplasmic reticulum. So this is the major difference between them. Is that clear? Okay, question 15. In a disease, androgens are produced, but the cell fails to respond to so androgen is a form of steroid hormone. So the cell fails to respond to the steroid hormones because they lack the appropriate intracellular receptor. After binding to the intracellular receptor, the steroids regulate the rate of which of the following? A. Yes, excellent. The transcription of genes. So this is important to know that the steroids mean of regulation of gene expression. Steroids, they are hydrophobic, meaning that they can diffuse into the cell membrane because they're fat soluble. They can diffuse into the cell membrane and then go into the nucleus, bind into the zinc finger DNA binding proteins, and then causing the transcription of genes. So they regulate mainly genes transcription. Steroids are the same as cortisol. Androgens are all forms of steroid hormones. Exactly, through zinc fingers. Excellent. Question 16. Which of the following is the smallest carbohydrate? A, D. Yeah. So the answer is D, glyceraldehyde. Anyone knows why? Yes, a triose. Exactly. So the smallest carbohydrate is glyceraldehyde because it's a triose. So here you can see in this diagram the monosaccharide structure. Monosaccharides, they can be either a triose like the glyceraldehyde or the dihydroxyacetone, or bentose like ribose and ribulose, and hexoses like glucose, galactose, and fructose. So the smallest ones are the trioses, either aldoses or ketoses. Glyceraldehyde and dihydroxyacetone. Triose meaning that they have only three carbon sugar. Okay, question 17. A 6-year-old African-American male is brought to your office for a routine checkup. His mother remarks that he often seems uninterested in playing with his beers and appears to run out of breath quickly. His medical records reveal that he has missed several pediatric vaccinations, and he has been hospitalized twice, one with a chest infection and one with an abdominal pain. The patient mentioned that occasionally his bones hurt. Which of the following protein changes most likely accounts for this patient's condition? A, glutamic acid substitution for valine. B, valine substitution for glutamic acid. C, phenylalanine substitution for proline. D, valine substitution for lysine. E is early termination of polypeptide synthesis. A. Correct. The diagnosis is sickle cell disease, but it's quite confusing how the choices are written. It is when we say glutamic acid substitution for valine, that means that glutamic acid is substituting valine. So B, we mean valine is substituting glutamic acid. The correct answer would be B, because in sickle cell disease, we have the substitution of normal hemoglobin have the glutamine, and in the mutated hemoglobin, they have valine. So valine is substituting glutamine. Is that clear? Yes, exactly. So all of these were symptoms of sickle cell anemia: exertional dyspnea, pneumonia, resulting in acute chest syndrome, and bone pain are clinical features of sickle cell anemia. It is a point mutation caused by valine to substitute the glutamic acid in the 6th position of the beta globin chain of hemoglobin. Okay, so it is the valine substituting the glutamine. Okay, for amino acids with neutral R groups, at any pH below the BI of an amino acid, or the BK, the population of amino acid in the solution will have what? A net negative charge, a net positive charge, no charged groups, no net charge, positive and negative charge in equal concentrations. Oh. B, exactly, a net positive charge. Anyone knows why? A cationic state. Perfect. Yes. So, yes, exactly. So these are 3 important things to always remember when it comes to the amino acid titration curve and the buffering system. So if the BH is equal to the BKA, then the A minus, which is the deprotonated form, or the negative form, is equal to the HA, which is the protonated form. And if the BH is more than the BKA, so we have more A minus, which is less protonated form and more of the deprotonated form. The deprotonated is the -1. And pH, higher pH, meaning that we are toward the alkaline side, as you can see on the graph. In our situation, we said that the pH is lower than the pKa. So we have more of the HA and more protonated form. So we have more of the positive charge. Yes. Is that clear? Okay. Moving on to the next question. When the malarial parasite infects an RBC. Okay, I included these two questions. I didn't come across them in the lectures, but I found them in the concepts and they didn't seem to be correct. So I included them for clarification. When the malarial parasite infects an RBC, its metabolic waste product results in a decrease in intracellular pH. Which of the following describes the consequences of the acidification of the hemoglobin? D. Yes, perfect. That's correct. There will be a right shift of the oxygen dissociation curve for hemoglobin. Okay. So I'm going to discuss I'm going to come across the second question and then we'll discuss them together. So for this graph, a person moves from the sea level to the mountain. What change will happen to the oxygen dissociation curve? C, yes, because of 2,3-bisphosphoglycerate, that's correct. Yes, excellent. So the answer would be C. This is the really important concept, although it's very confusing, but it's really important to understand the oxygen dissociation curve, the right shift and the left shift. So to explain this, the right shift is always think about it that oxygen is needed. So we need to unload oxygen from the hemoglobin. In that state, hemoglobin is going to be in the taut form. In the taut form because it has low oxygen affinity. So it is unloading oxygen. So what causes the right shift of the oxygen dissociation curve? Exercise, because we need to give the tissues, the muscles more oxygen. If we have a decrease in pH or the increase in hydrogen ions, an increase in temperature, and in cases of hypoxia, as the previous case where we have high altitude, it causes an increase in 2,3-bisphosphoglycerate, which causes the right shift of hemoglobin. So always think of the right shift that we need to lose oxygen. We need to unload oxygen from hemoglobin to give the tissues, okay? Now the left shift is opposite. Oxygen is sequestered into the hemoglobin. So when we shift the curve into the left, that means that hemoglobin is holding tightly into the oxygen, and this can occur physiologically if the body is having decrease in oxygen needs, such as if decrease in temperature, because if the body has decrease in the temperature, that means that most of the organs are not metabolically active, and thus less oxygen needed to be released into the tissues. And physiologically also in the fetal hemoglobin, because they have a decrease in levels of 2,3-bisphosphoglycerate. Now, pathological left shift can occur if we have something that holds up into the makes the oxygen to be sequestered into hemoglobin, such as carbon monoxide and methemoglobinemia. So that prevents the oxygen to be delivered into the tissues, like carbon monoxide and methemoglobinemia. Okay, is that clear? Okay, perfect. Moving on to the last question. Okay. The mother of a 3-year-old boy is referred to genetic counseling after her son is diagnosed with an enzyme deficiency. Recently, the mother noticed that her son has an abnormal facial appearance and mental retardation. Upon eye examination, his corneas are clear bilaterally. The genetic analysis revealed an X-linked disease. What is the enzyme that is most likely to be deficient in this patient? C. That's correct. The L-iodronate 2 sulfatase. Perfect. Corneal clouding occurs in the alpha L-iodronase, which is seen in the Hurler syndrome. Now for A, the sphingomyelinase, it is the enzyme which is deficient in, anyone knows? It's not covered in this part of the review, but so sphingomyelinase deficient in Niemann-Beck disease. It presents with the cherry red spots on the retina, as well as hepatosplenomegaly. And hexaminidase A, it is the enzyme deficient in Tay-Sachs disease, which also has the cherry red spot, but no hepatosplenomegaly. So all of these choices they have and manifestations in the eyes, but the C has no manifestation, no corneal clouding, only mental retardation and abnormal facial appearance. So the answer is C, L-iodronate 2 sulfatase. So this is from your slides, the Hurler and Hunter. I don't think the last two, the Sanfilippo and Morchio, are not really important to focus on. The most commonly tested are the difference between Hurler and Hunter. Both of them cause the accumulation of dermatan sulfate and heparin sulfate, but the key difference in Hurler syndrome is the corneal clouding. The presence of corneal clouding, along with dystosis multiplex, which are the skeletal deformities, organomegaly, and mental retardation. Hunter, however, has no corneal clouding, and it is X-linked. So this is important to note that Hunter is X-linked. They usually have this mnemonic where the Hunter, he sees the target clearly and he points, the target is X. So that's to remember that Hunter, they are seeing clearly, they have no corneal clouding, and they're X-linked. One important feature that can help you also in other scenarios, that Hunters are usually described as being aggressive, being severely mentally retarded, so that they're aggressive, okay? And it makes sense, like Hunters and aggressive, so you might think about it like this. Yes, organomegaly occurs in most of the mucopolysaccharidosis and as well as the lysosomal storage diseases, glycogen storage disease. So they're not a specific thing to look out for. Try to pick in these glycogen storage and mucopolysaccharidosis, look for these specific findings. Like in Hurler, it is the presence of corneal clouding, as well as the dystosis multiplex, the skeletal deformities. In Hunter, there is no corneal clouding, X-linked recessive, and mental retardation. The last two, the Sanfilippo and Morchio, they have no specific findings. Like, I don't think, I never came across any question from them, honestly. But read them for the sake of completion. Okay, I'm done with the questions. Any questions from you guys? Thank you so much. I hope that was beneficial. And if you have any question, my email and my phone number are in the slide, so you can text me even if it's not on the lectures included. Text me at any time and I will be happy to assist. Thank you so much, guys. Could you please scan this for your feedback? I would really appreciate this. Thank you, guys. Wish you best of luck in your exam.
Thank you so much.
Thank you.